Hello, my name is Emmanuel Van Acker. To start, let's all have a walk on the beach and take a good deep breath. Can you smell the sea? You have just inhaled a good portion of the sea spray aerosols, containing biogenic chemicals, so-called marine bioaerosols. But what are marine bioaerosols, and which effects could they have on human health? When waves break, they produce air bubbles that reach the surface and burst, launching tiny water droplets into the air, forming sea spray aerosols. These sea spray aerosols are capable of transporting all kinds of biogenic chemicals. In this way, coastal populations are exposed to nearly all chemicals produced in the sea. Biogenic chemicals we're investigating in case of sea spray aerosolization are algal toxins or phycotoxins. These chemicals are produced by harmful algal bloom species and are known to be noxious via the consumption of contaminated seafood. For the exposure via sea spray aerosols, there are already a few reported cases where people have a severe irritation at the respiratory tract during the aerosolization of two toxins, brevetoxin and ovatoxin. On the other hand, there is a new hypothesis which was recently put forward to explain the improved health of coastal populations. The biogenics hypothesis states that this improved health is a consequence of a regular exposure to low concentrations of biogenic chemicals like these phycotoxins in aerosols. But we want to know, holds this true also for phycotoxins? We investigated the effects of aerosolized phycotoxins, exposing two epithelial lung cell lines to several phycotoxins in 2-day in vitro experiments. Adverse effects at high concentrations were examined using viability assays, and to examine positive effects at low concentrations, we analyzed the activity of the P-idrica-act-mTOR kinase pathway. The downregulation of this specific pathway suppresses pathological conditions such as cancer, diabetes, and inflammation. Looking at the viability results in function of the toxin concentration in the air, we see that the azotoxin, so the red data, has an adverse effect at much lower concentrations compared to oxalic acid and brevetoxin. This is rather surprising, since azotoxin is generally seen or considered to be one of the least potent phycotoxins. If we compare these effect concentrations with the measured aerosolized concentrations of phycotoxins in natural conditions, meaning actually brevetoxin and ovatoxin during a harmful algal bloom, here depicted in a gray color, the effect concentrations of azotoxin have the same order of magnitude. However, we're also interested in lower concentrations, possible ambient concentrations even below the current limits of detection, here depicted in a blue-green color. To look at the downregulation of the mTOR kinase pathway, shown here on the left y-axis, we want to control. We want to compare with a positive control. This product, PP242, is a specific kinase inhibitor for this specific mTOR pathway. The phycotoxin we found to have a similar effect is azotoxin. Starting from a concentration which is 10,000 times lower than the adverse effect concentrations, we saw a downregulation of this pathway between 40 and 50%. To be clear, this is a positive effect compared to the adverse effects at high concentrations, so the adverse effect on viability. To conclude, at low concentrations, aerosolized phycotoxins are rather friend than foe. If confirmed by subsequent testing, these results are the first supporting the biogenics hypothesis. And I still brought a small present for you guys from the sea, some miracle elixir of life.